I'm Ada. I recently graduated high school and I'm an avid researcher in genetic engineering. And in this video, I would like to walk you through the insights I learned from writing a review article on optogenetic CRISPR-Cas9 tools using two methods called light-induced dimerization and photochemical control. I hope you enjoy this video and have some key takeaways at the end. To write this review, I combined my two main interests, which is optogenetics and genetic engineering. But if you know about optogenetics, you know that it uses gene editing to make cells light sensitive. But then, how do you use that technology to make the main tool of gene editing, that's CRISPR-Cas9, itself optogenetic? So, turns out gene editing is not the only way to make cells light sensitive. There are also other molecular ways that we're going to see through this article. Often, great discoveries are made when two diverse fields combine to extend their capabilities. Optogenetics, which was originally made to control neurons, has joined forces with the most versatile tool for gene editing to exist yet, paving the way for new treatments and discoveries. Before getting into the technicalities of the methods used, let's first explain what is in optogenetic CRISPR-Cas9. So essentially, it is a light-sensitive CRISPR-Cas9, meaning when CRISPR-Cas9 is exposed to the right wavelength of light, it can become active and conduct gene editing through methods such as NHEJ and HDR. And when the light wavelength is switched off, it becomes inactive. So it provides us with an off on switch to control CRISPR-Cas9. But you would think, why does this matter? The problem with current CRISPR-Cas9 is that sometimes single guide RNAs can lead to off-target effects. This can cause double-stranded breaks in untargeted regions. This can cause adverse consequences such as gene mutations, insertions, deletions, and even leading to cancer. Thus, we need to find a more precise way to conduct gene editing through CRISPR-Cas9. In the past, people have used chemical methods to precisely control the modifications made by CRISPR-Cas9. An example is using doxycycline regulated Cas9. However, we've seen adverse consequences through these methods as well. Chemically induced CRISPR-Cas9 have potential for inducing cytotoxicity. Doxycycline can even affect cell formation and colony formation. These inducer chemicals can actually diffuse freely, so this limits the spatial resolution of where CRISPR-Cas9 is active. It's difficult to remove these chemicals, and this prevents us from being able to control Cas9, CRISPR-Cas9 precisely, and prevents us from turning it off and on quickly. If these are all the disadvantages, then why is optogenetics a better alternative? So, light is a reversible and non-invasive inducer modality, meaning that we can precisely turn CRISPR-Cas9 on and off non-invasively and also control its spatiotemporal precision. This can prove to be a way to tackle all the disadvantages such as off-target effects which we face with CRISPR-Cas9. So this is the review paper currently available on Medium. This review I have looked at the development of optogenetic CRISPR-Cas9 tools for the purpose of gene editing through NHEJ and HDR. So I have analyzed them on the metrics of analyzing their molecular constructs, the unique capabilities that have so far been demonstrated, and the limitations that they pose, and what kind of implications do they have for the future in both medical sciences and for furthering our research techniques. This is the table of contents that is covered in this review, and in this video, I'll be giving you a high level summary of what I've learned through this. I have analyzed two types of methods that can be used to make CRISPR-Cas9 tools light sensitive. The first one is called split systems, or it's also called as light-induced dimerization. As the word suggests, you're splitting something, right? And in this, a single protein is divided into two or more separate fragments. And when these fragments come together, and that is usually done through the exposure of light, they can reconstitute the functional protein and create and become functional. Two proteins can become the two fragments of proteins can come together through the formation of covalent bonds and become functional once again. Also gives us an off and on switch because before light is before the CRISPR-Cas9 is exposed to light, these two fragments are separate, and on exposure they come together and then they become functional. So we can decide to when to make the CRISPR-Cas9 function. The method I have focused on is photochemical control. So it's refers to regulation of chemical processes using light as an external stimulus. Basically, what we're doing is we're using light to induce specific chemical reaction or trigger molecular changes in a controlled manner. This also gives us the same off-on switching and also reversibility. So you take the light away, the CRISPR-Cas9 goes back to its non-functional mode. Based on this, I have totally analyzed five tools, so two under split systems and light-induced dimerizations, and three under photochemical control. The first tool is called PA-Cas9, which stands for photoactivable Cas9, in which Cas9 is broken into two fragments, and these two fragments they are known as N fragment and C fragment, which are attached to a photo-inducible dimerization system that was developed known as magnets. Fragment is attached to PMAG and C fragment is attached to NMAG, and on exposure to blue light, it reconstitutes a functional Cas9 which can induce double-stranded breaks at the targeted DNA, which can thus and thus NHEJ and HDR mediated DNA repair can occur. Main capabilities apart from conducting gene editing through both the methods is that uh, frequency of indel mutation increases with the exposure of light. It can also act as a photoactivable nickase, and it offers spatial control of gene editing. So, gene editing will only occur on the parts where you expose the light, and it's also reversible. So, if you stop the exposure of light, uh, exposure of Cas9 to light, the gene editing will thus be stopped as well. And all the observations for these different claims have been detailed in the review paper. So if you are really curious of like what is the evidence behind these claims, you could definitely go through this. Some limitations are that there's still a little bit of background activity present in the absence of light. Sometimes editing can be done even when light is not, the CRISPR-Cas9 is not exposed to light. There's also a limited spectrum of light that you can expose Cas9 to. And blue light has been observed to show cytotoxic effects sometimes, and also it cannot penetrate the biological tissues deeply. So you can only target surface level tissues through this. The future implications of this is that it can definitely reduce off target effects to a huge extent and also expand the use of viral vectors. So, Cas9 applications have been limited due to packaging size, limitation of viral vectors, but because the DNA fragment of PA-Cas9 is shorter than full-length Cas9, it will be possible to separately package these two fragments into viral vectors, which can expand what kind of viral vectors we use. The second system is called FAST system, so it actually stands for FRL activated split Cas9 system. This also uses a similar method, so Cas9 is broken into N and C fragments, and it is attached to photoinducible dimerization system. But in this case, it is derived from the dimerization system is derived from the bacteria known as Clostridium thermocellum. The expression of this fragment is driven by the kinetic promoter known as PFRL, and the expression of this uh, complex is driven by the constitutive promoter PHCMB. The main difference between this system and the previous one is that it uses a different light wavelength, which is far-red light. The previous one used blue light, and there are several benefits because of this choice. First is that it has lower photocytotoxicity than blue light and UV rays. So far in the study, there were no off-target effects, and also the depth that can be reached through far red light is greater than the one through blue light because of the difference in wavelength. So in this study, they had gene edited uh, livers of mice as well as other internal organs. And it also was shown as a potential for anti-cancer therapy. While these capabilities sound great, we do have to keep in mind that this paper only presents an initial proof of principle. Go over to the second method, that is photochemical control. So the first tool we're going to see is it uses a caged lysine amino acid to build light activated Cas9. So that's a lot of words, but what does it actually do? So, lysine photocaging is a method in which uh, a covalently bound photolabile protecting group, so a photocage, is introduced on top of a protein or ligand. So this forms covalent bonds with it, thus forming a cage and rendering the system inactive. When you expose this system to light, these covalent bonds are broken and the photocage is separated, thus giving us back the functionality of the molecule. This we photocage Cas9 using lysines. So lysines are actually present in the amino acid sequence of Cas9 protein, but in this genome of human cells was expanded to recode the amber stop codon tag to, instead of terminating the protein encoding during translation, it incorporates caged lysines into the protein sequence of Cas9, thus giving us a photocaged Cas9. This is the molecular diagram of lysine. Photocleavable group is shown in red, and this is attached to Cas9. One of the key benefits is that there can be multiple possible caging sites, each with its own advantages and disadvantages. So this can give more freedom to researchers. The fourth tool that we'll look at is called light activated guide RNA. So in this tool we shift from modifying Cas9 to guide RNAs. So this also uses photocaging, but for guide RNAs. So guide RNAs are caged by attaching guanosine and uridine to NPOM, which is nitropyperonaloxymethylene. So the Cas9 systems, in which Cas9 is modified, they require protein engineering, and that includes like screening of mutations and split sites, directed evolution, and unnatural amino acid mutagenesis. But through chemically modifying guide RNAs, we can uh, circumvent that, and it can also provide us a direct path to regulate interactions between Cas9 and guide RNA, ribonucleoprotein complex, and the target double-stranded DNA as well. So it gives us more freedom, which the Cas9 systems were not able to. So four bases that are evenly distributed in the 5' photospacer regions are caged, and this suppresses the target hybridization between the guide RNA and double-stranded DNA. This is the molecular diagram of the caged uridine and caged guanosine. Distinct advantage that this provides is that it has increased guide RNA stability. And this was actually the study was shown in both mammalian cells, including targeted at the human genomic locus within CT and NB1 gene, and it was also targeted at aquatic embryos. So this shows that this can have a cross-species application. This tool that we'll look at is CRISPR Plus. This also uses photocaging, but for guide RNAs again. Different approach. So in this, we designed protectors for guide RNA, which hybridize with sgRNA, and they detach in the presence of light. In this protector, oligonucleotides are made that are photocleavable and they're attached to sgRNA. When it's exposed to light, these fragments, photocleavable oligonucleotides break into fragments. These have a lower melting point, so they then have a lower affinity to sgRNA. This paves the way for sgRNA to hybridize to the target DNA, and then Cas9 can conduct or can induce double-stranded breaks, allowing gene editing repair methods to undergo. The main capabilities that it has is that we can target multiple DNA sequences through this, through using multiple guide RNAs that are photocaged, but they can target different genomic loci. So those were a lot of interesting facts about these various optogenetic CRISPR-Cas9 tools. If you would like to compare them at a glance, this you can pause the video and read through this data table that I've created, in which I compare them using some metrics such as which optogenetic method we're using, what kind of molecule we're modifying, the type of light required, as well as I've also mentioned the distinct advantage they have, what is the level of background activity that is observed, and some other key limitations that might uh, prove to be quite important when choosing which tool to use in a study. Now let's consider some future applications. So the FAST system, as I mentioned before, had was shown to have potential for anticancer therapy. So optogenetic CRISPR-Cas9 tools can prove as an effective medium to for these therapies. As well, because they can target these tumor suppressor genes and restore them to interdict the tumorigenesis. And they can also expand the techniques we use to study current genetic diseases because they provide greater spatiotemporal precision. So, optogenetic proves to be a powerful tool for scientists to control the activity of biological cells, as we've just observed. So, its application in CRISPR-Cas9 can help scientists reduce off-target effects by controlling the time and space in which Cas9 nuclease or sgRNA is active. But as with any new field, more research and technological advancements need to be done. So, some of the cases that need more research is like which safe and reliable light sources should we use to target deeper tissue areas. As well, we need to conduct longer studies to measure the long term effects on cytotoxicity and other metrics. So these CRISPR-Cas9 tools so far have only been shown in animal models or cell cultures. So we need to verify the therapeutic potential for cancer through clinical trials. But nevertheless, these tools do give us unprecedented control over the extent of gene editing. And the new studies and treatments which can be developed through the convergence of these two fields are exciting to look forward to. I really hope you were able to learn something new through this video. I would request you to still go through the review article I have written, and I would love to know your thoughts and if you have any feedback for me, as I would love to incorporate that and make more improvements to make it even more effective for people who are curious about this field. Thank you so much for your time. I'm Ada Chaudhry, and keep reading, keep learning. I hope you have a great day ahead.